Hi everyone, welcome to another episode of the tutorials on Bacterial Genome Analysis. And let me also say that the ebook as well as the script for this tutorial are all available on my Patreon channel. So check the description box for the link and then you can use that to get the materials. So take note of that. In today's video, we are going to look at genome assembly of Methicillin-Resistant Staphylococcus aureus using Oxford Nanopore data. And so we are going to reproduce this online gallery tutorial. I will leave the link to this page in the description box. And if you're a beginner, especially, then I will encourage you to read the introduction part here. It's very important. Now, antimicrobial resistance is a major global concern because it reduces the ability of health professionals to treat infections. And this leads to a lot of deaths, thousands of deaths yearly. And so it's important to understand the processes involved, and this can help to design intervention measures, which includes drug development as well. And so one way, or let's say one approach that is used, which is effective by the one approach that is used to study antimicrobial resistant genes, is through genome sequencing. And so after generating your data, you now have to use these computational tools to derive insights and then tell your story. And this is where this tutorial comes in handy. So this tutorial will show you some tools. Okay, it will show you the steps that are involved in analyzing bacterial genomes to detect antimicrobial resistant genes. So that's the focus of this tutorial, by the way, to look at an antimicrobial resistant organism and then look at its characteristics in terms of the genome sequence. So that's what we are going to do now. So for this tutorial, we are going to follow the steps here, and there's a breakdown. We are first going to install the tools we will use for this exercise, and then we are going to download the example data. Then we will perform quality control and read filtering, and then we are also going to perform genome assembly and visualization. After that, we will look at plasmid detection and then antimicrobial resistant genes detection. And we're also going to look at genome annotation and visualization. So these are the activities we are going to perform. So that's it. Now, if you want to follow this tutorial, you need to make sure you have a Unix-like operating system. And so I recommend Linux. That's what I use for my tutorials, by the way. And so I'll encourage you to also use Linux. It's possible to use macOS, but I prefer to use Linux because it's more flexible. I prefer to use Linux because it's more flexible. You should also have Mamba or micromamba installed. We are going to use Mamba or micromamba to install the tools. So I have tutorials that show how to install them. So check the description box. You find a link to the video. So use those videos to install Mamba or micromamba and then set up your PC. So that's it. So let's get started. So let's open the terminal and start the exercise. So I'll open mine now. I am on the terminal now. So what I'm going to do next is to install the tools. So I'll use Mamba. You can also use micromamba to install. Again, instructions for installing Mamba and micromamba can be found in the description box. All right. So I'll begin by creating an environment. So I'll say mamba create-n, and I'll say bacteria. This is the name of the environment. So let's execute this command. All right, the environment has been created. So I'm going to activate it using this command. If you created it using micromamba, then you have to specify it here in place of this. All right, so let's activate the environment. So I'll say mamba activate bacteria. So let's execute this command. All right, the environment has been activated. So what we have to do next is to install the tools. So to install the tools, I'll say mamba install, and I'll indicate the channels. So I'll say that C, bioconda-C, conda forge-C, defaults. These are channels, and then I'll specify the tools. So I'll use Flye, this will be used for genome assembly. Filtlong will be used for filtering the reads. We will also use Bandage to visualize the assembly. We will use QUAST to evaluate the genome assembly. We will use NanoPlot. All right, like this. Let me just redo it here. So from Bandage, I will say NanoPlot. All right, so NanoPlot will be used for quality control. And then QUAST, this will be used for evaluating the genome assemblies. We will also install PROKKA. This will be used for genome annotation. We are also going to use STARAMR. This will be used to identify the antimicrobial resistant genes. And then we're also going to use Minimap 2. This will be used for mapping the reads. We will also use some tools. And then we will specify these Python packages, BioPython, and then bcbio-gff. So these are the tools we will install. All right. So let's execute this command to install the tools. All right, so Mamba has started its way. So let's wait for everything to get done for us. So let's type Y to confirm and let's hit the enter button. So now Mamba is going to download all the necessary packages, install and configure them for us. So let's wait for everything to get done for us. All right, now all the tools have been installed except one, but we will install that later on. So for now, let's clear the screen and continue. I repeat, there's one more tool that we install, but we will do that later on in the tutorial. So let's continue. So what we are going to do next is to create a working directory and then cd into it. So I'll say make the MRSA. You can use any name of your choice, it's fine. Or you can also say bacteria, that's fine. All right, so I've created the working directory. So I'll cd into it. So I'll say cd MRSA. Nice, that has been done. So now that we have created this working environment and cd into it, we can now proceed. So what we are going to do next is to download the example data. So let's go for the link. So on the Galaxy page, just scroll down, or you can just use this site. So when you are here, you can click on quality control. That will send you down. So on the Galaxy page here, just scroll down a bit, and you are going to find the section which has the down link, and that is this side here. So what we are going to do is to get the download link. Now with the downloads, if you want to download using your browser, you can just highlight and then let's say right click and then go to open a new tab or whatever. So once you open it, the file will be downloaded, but I will do it on the terminal. So what I'll do is to copy this link. So I'll highlight, I've already done it here, and then I'll copy it. There's also this button here to enable you to copy. So any of them should be fine. So just make sure to get the download link. So let's go back to the terminal. And then let's download it. So I'll use wget, but you can also use kill. So I'll say wget, and then I supply it with the download link, which is this one here. So I'll execute this command now. So this will download the file for me. So let's wait for everything to get done for us. Okay, the file has been downloaded. So let's do an ls. We are going to find it there. So this is it. So with this file, it's compressed, so we have to decompress it. So we are now going to decompress this file here. So let's just do it. So with the decompression, we are going to use bzip2 to do it. So I will say bzip2, and then I'll specify this flag. I'll say -d, and then I'll specify the file. Okay, so this is how I'll do it. So let's execute this command. So this will take some time, so you should be patient. Okay, the file has been decompressed. It took about 1 minute on my end, but it can take more depending on your system, the PC you are using for the exercise. So let's continue. So let's do an ls, and we are going to find the file there. So this is the file. So there's a FASTQ file. So now we have the data, so we can start the analysis. So let's clear the screen and continue. All right, so let's do an ls to take a look at the file again. There's the file. Now the file we have here, what you should take note is that this file is uncompressed. Normally, FASTQ files come in a compressed format, i.e. the .gz format. So that helps us to save space. But for this tutorial, we are going to use the uncompressed one here. But if you have your file, make sure that it's compressed. That's what I recommend so that I can save space. All right. What we are going to do next is to perform quality control. So with the quality control, we are going to do 3 things here. We are first going to assess the quality of the raw reads, which can be found in this file. And then we are going to filter the reads. And after filtering, we are going to assess the quality of the file that we get after filtering, the filtered reads. We are going to assess the quality. And then we are going to compare pre and then post filtering. So I mean that we are going to compare the raw reads to what we are going to get after filtering. So that's what we are going to do next. So let's first create a QC report output directory. So I'll say make the QC_reports. Yes, perfect. So now what I'm going to do next is to assess the quality of the raw reads. And so I'm going to use NanoPlot. So to use NanoPlot to assess the quality, we say NanoPlot, we call it, and then we specify our FASTQ, which is this one. So we have indicated the name. And then we also need to specify this flag to show the N50 mark. So I'll say -N50. And I'll say -O. And I'll say QC_report/ and then I'll paste the same sample name there, like this. And this time I'll say _raw. This is because we are going to filter the reads and then we are also going to perform QC. So I want to differentiate between the QC report for the raw reads and then the filtered ones. I will do that later. All right, so let's execute this command. Okay, so this command, when I executed it, took about 1 minute. So you should also monitor the time. It will take maybe around the same time to complete. So let's wait for everything to get done for us. All right, QC has been performed. So let's do an ls. We are going to find our directory here. This is what we created first, and inside it we have the results. So I'll say ls QC_reports, and then I'm going to see this directory. So this directory has the output files. So I'll do ls again QC_reports/dir, etc. And I'll display the content. So I have them here. So we have plots, we have HTML files, we also have text files. So we are going to look at some of them quickly and then proceed. So I'll do that using my file manager. All right, so these are the output files. So we have images, we also have HTML files, and we also have text files. So I'm going to focus on just these two files, this and this. So this one has the summary. So you can open it with any text file editor. So that's it. So we have some summary here, and then this one has this file here. Let me just go over. And this file here has all the reports in a single HTML file, including the figures here or the plot. So you can also open it with your browser like this, and you will see all of them there. So you can do that and also do some comparison. Now for this tutorial, I'll focus on just this summary here. I'll make tutorials later on which goes into details with the UC reports. But for now, let's take a look at just the summary. So the summary here gives you an overview of your data. So we have, let me just enlarge it a bit if it's over. Yeah. So we have general summary, mean read length, mean read quality, median read length, read quality, median, etc. So it goes on in that order. We also have top 5 highest mean base core quality scores and their read length. We also have top 5 longest reads and their mean base quality score. So these things give you an overview of your data. So it can help you to get the average of your, or let's just it can give you an idea of the length of your reads and also the quality. So with the long reads data, if you're comparing to short reads, you realize that the long reads are of lower quality than the short reads, which is to be expected. That is what happens, by the way. And so you shouldn't be surprised if you compare this to some short read sequences. This one will have lower quality, and this is where the other step come in. So what we are going to do next is to filter the reads, and then hopefully we should see some improvements. We are going to do that and we are going to compare. So after filtering the reads, we are going to perform the QC again. Then we are going to compare the metrics here, the summary here, or the metrics here. We are going to compare the one for the raw data, that's the raw reads. That's what we are looking at. You compare with the filtered reads. That's what we are going to do next. So that's what we'll do. So with that, we'll still use NanoPlot. So the guide tutorial also did it here, by the way. So what we are going to do is to check and then just do something similar. So all the results are here, by the way, but we are going to do the command line version as well and then compare to see if we have a similar observation. So that's what we are going to do next. So let's go back to the terminal. So let's clear the screen and continue. All right, so let's do an ls to look at the file again. So we have it here. So what we are going to do next is to filter the reads. So we are going to remove reads with lengths shorter than 1000 BP. So we have the instructions here. Let's go back to the Galaxy page. Yeah, so we are going to use this tool, filtlong. We have already installed it, by the way. So from the instructions here, we have minimum length, that's 1000. So what we are going to do is to run filtlong and then also specify the min length, and then filtlong will remove all reads that have their length shorter than 1000 base pairs. So let's go back to the command line. All right, so we will call filtlong now. So I'll say filtlong, and then I'll say dash dash main underscore length, and I'll say 1000, and then I'll specify the raw file, and then I'll indicate this. So this one, we are going to output this to a file. Now see, I'll indicate the file name again. So it's DRR 187567. Let's just make sure that is it. So we have DRR 187567. And then I'll say dot filtered dot FASTQ. So we want to differentiate this new file from the old one. So that is why here I have dot filtered. So the dot filtered is also a description. So even if someone was not involved in the process, once the person sees this name, the person will have an idea that there was some filtering done. So that's why it's good to have a good naming convention. All right. So let's execute this command to filter the reads. All right, filtering has been done. So let's do an ls, and we are going to find that file also here. So this is the new file, the filtered reads. So what we are going to do next is to assess the quality of the new data we have, the filtered reads. So we will use NanoPlot again. So it's the same command, but this time we are going to use this file. So we'll call NanoPlot now. So to assess the quality of your data, the FASTQ file that we have, the long reads file, you will say NanoPlot, and then we will say dash FASTQ, and then we indicate the FASTQ. So it's going to be this file. So I'll say drr.filtered.FASTQ, and then I am also going to indicate a flag which shows the N50 mark. So I will say dash N50, and then I'll indicate an output. So I'm going to save it in this directory also. So I'll say -o, and I'll say QC_reports / and then I'll indicate the name here. So I'll just paste it, and then I'll indicate another thing. I'll say _filtered, so that we will know that this is the QC report for the filtered reads. So let's execute this command. All right, Nanopore has completed its work. So let's do an ls into the QC_report directory, and we are going to find our directory there. Yeah, so that's it. So that's the output. So you can ls into it also, and then check the files out. So we have them here. So we are going to also check these files using our file manager. So let's do that now. All right, so we have the output here. So again, you can check the summary statistics in the test file, but I'll use the HTML file. So I'll open it now. So I have that also here. So what we are going to do is to compare what we have in the new output, the new result, that's for the filtered ones, to this one here, and then see the outputs. So let's do that. The guide tutorial has already combined those two reports, so we can find them here. So this is what we have. Let's even check ours. Let's see. Yeah, okay. Yeah, so we have them here. So you can compare them. So let's compare. And we also have some questions and then some solutions here. So these questions are about the reports here. And so I'm going to check them and then just read them. So we have this first question. It says, what is the increase of your median read length? So if you compare, there's the read length, let's just check. So there's a 60.3% increase. So let's take a look at the median read length. So there's the median read length. So we have not filtered, that's a raw one, and then the filtered, which is this one here. So in this tutorial, not filtered, I put it as raw. So I'm using the word raw and then not filtered respectively with respect to the information here. So take note of that. All right. I repeat. So whenever I say raw data, it's the same as not filtered in the tutorial here. So take note of that. All right. So let's look at our report. So mine is here. So there's the median read length for the raw, that's the not filtered, and then for the filtered, we have that also here. Let's check, which is here. So that's what the tutorial here reports as the 60.3% increase. So that's what we have. So that means there has been an improvement. So median read length, the change is 60.3%. So from 3,400 to 5,451. Yeah. So that's what we have here. Okay. So that's for the median read length. There are other questions here. It says, what is the decrease in total bases? That one too, we have the answer here. It says minus 2.0 decrease. So it indicates that it's not a very significant decrease. So our data was quite good to start with. So that means even though there was a decrease, it will not really have an impact on our results because the change is very, very small. It's not significant. So that's basically what this implies. And then we also have what is the coverage. So the coverage here is a measure of, on average, how many reads cover a single base in the genome. And so what the guide tutorial here tells us is that if you divide the total reads, so here the total reads will be the total length. So with a FASTQ file, you will have several reads. So for each of the reads, you have a length. So if you combine all the lengths together, if you sum up all the lengths, that becomes the total read. That's the tutorial here is making mention of. So if you divide the total reads, that is the total length, by the genome size, then you will get a number of how many times your genomes could theoretically be covered by reads. That is what this statement simply means. And the computation has been done. So for the raw data, the unfiltered one, this was the total length, and then divided by the genome size, we get 214.4. And then after filtering, we get this. That's what we have here. So this statement also lets us know that comparing the coverage for the raw data to the filtered one, there's not much of a big difference in coverage. And so this means that we shouldn't be worried about the downstream analysis with respect to the coverage, because before filtering and after filtering, the difference is not that much. It's just this 214.4, this is 210.2, so it's just a little difference. So we shouldn't be worried. So basically, that's how we do it. So for the coverage, it's possible to compute it. You can use Python R or even Bash. I've tried with Bash, by the way. So it's possible to do that. So try your hands on that and let me get your comments or feedback. If you also want to do that later on, you can always contact me and then we can arrange a session to do that together. So that's it. So let's proceed. So we will move on to the terminal to continue the analysis. So we are now done with the quality control section. So we'll move on to the assembly part. So we are going to assemble the data. So with the assembly, we are going to use the filtered read. So the instructions are here. We are going to use Flye. I've covered some tutorials on Flye, by the way. So this is going to be an add-on. So we are going to perform the genome assembly using Flye, and the input is going to be the filtered reads. So let's go back to the terminal and then continue. We have the genome size also here. So Flye allows you to specify a genome size. So in this tutorial, the genome size that was used was this one, 2.8 M. That is what was used. So we will also use the same. So let's go to the terminal and then continue. So let's clear the screen and start. All right, so let's do an ls to take a look at our file again. So this is going to be our input file, the one we filtered. So this is methicillin-resistant Staphylococcus aureus data. So we are going to assemble this data. So that's what we are going to do next. So we are going to use Flye because this is a long read data, ONT read. So we are going to assemble it. So to assemble with Flye, we will say Flye. And then we are also going to indicate some extra information. We need that for the assembly. So we will say dash dash nano dash raw because this is a nanopore data. And then we will specify the file. We are using the filtered one. And then we are also going to indicate something. So we say dash gene to indicate the genome size. So with Staphylococcus aureus, we'll use the genome size 2.8 MB. So I'll say 2.8 M. We are following the tutorial on the Galaxy page. So on the Galaxy tutorial page, the genome size was 2.8 MB. So that is what we are also using. All right, so let's get back to the terminal. And then we're also going to indicate the number of threads. So dash T, I'll say 8. So number of threads, you need to indicate it. And so if you have a high NPC or if you're using a Linux server, a cluster, you can increase this number to 10 or 20 or whatever. That will also make the process faster. The genome assembly process is computationally expensive, so ideally you should have a high-end PC that can do the heavy lifting basically. So for me, I'm using my normal laptop. That is why I'm specifying 8. But if you have more threads or more cores, you can increase it. That's fine. All right, let's also indicate something else. You will say -O to specify the output directory. So I'll say assembly. You can use any name of your choice. That's it. So that is what we are going to use. So with Flye, it also allows you to do polishing. So if you want to do some polishing, you can add a -I and specify the number of rounds, maybe 1, 2, or 3. 1 or 2 or 3. And that's up to you. But for this tutorial, we are going to use the default settings with respect to the polishing because I want to follow the instructions that has been given in the Galaxy tutorial page. So here, that's what they had. They didn't indicate the polishing option here, so I'll leave it. Maybe another time I'll include that. All right. So this is the final command to assemble our data. So let's execute this command. Okay, so the genome assembly process has started. So we have to wait for everything to get done fast. So it can take some time. So just be patient. And then depending on your computing power, it also affects the time it takes to complete the genome assembly process. So you should be patient and wait for everything to get done for you. Okay, genome assembly has been done. So this will help you get started. This is a summary of the results. So we have the mean coverage here that has been given here. So remember on the tutorial page, we had something similar being given to us as the mean coverage. So this has been reflected here. Okay, so let's proceed. So now let's do this. Let's clear the screen and then continue. All right, so let's do an ls, and we are going to find our assembly directory. So this directory has the output files. Let's ls into it. We are going to find some files here. So I'll focus on a few of them. So this one here, Flye.log, this has the logs. Remember when you started running Flye, it shows some text, it displays some text. So all those information can be found in this file here. You can look at it. We also have assembly-info.txt. We will look at that later on. We also have assembly graphs. We have this. We also have this, and we also have assembly.fasta. This fasta file contains the contigs. Now let's take a look at what was done after assembly. Let's go to the gallery tutorial page to see. All right, so from the page here, we see that after running Flye, we also have to take a look at the assembly info file. So for this tutorial, the developers or the writers, this is what they got. They had this table. You have sequence name, length, coverage, etc. And then from here we see two contigs. That's what they got. Now let's also go to the command line and then take a look at the assembly-info.txt file. So we will see what we get. So it's a text file. So we will just say cat assembly/assembly_info.txt. Let's execute this command. All right, so that's what we have. So we also have some results here. So we have 3 contigs, contig 1, 2, and then 4. But when we take a look at the tutorial page here, what was shown are 2 contigs. And we have this note here. It says the results can differ from this example because Flye can differ a bit per assembly. So that is what we have seen here. So we have 3 contigs, but here it's 2. So that means that this statement here that has been indicated has reflected in our results. So all right, so let's take a look at something else here. So we have our table here. Let's see. We have 3 contigs. So that's what we have. So contig 1, there's the length. We have contig 2 and then contig 4. So they also have contig 1 and 2. These are the respective lengths and coverages. All right. So these things will help you get started. It's important you always look at that. And then you also have to factor in the genome size. You also even have to factor in if the organism that you are dealing with has plasmids or not. I mean, there are certain things you also need to factor. I mean, the biology of the organism. So some of these things, once you know them, it helps you to easily understand the results you have. So we are going to proceed. All right. So with the genome assembly, after doing it, it's possible to visualize. So we can do that by looking at the content in the assembly graph. So there's the assembly graph. So we can visualize it. Let's check the gallery tutorial page and see what was done. So that's what has been given here. So we have bandage image. So this section here shows us how to visualize the genome assembly. Okay, so that means we have to use the assembly graph. So we have to also use Bandage, which we have installed, by the way, and then we will supply it with our assembly graph. That one too, we will do that. I'll show you everything on the terminal. So let's do that and then compare the outputs we have to this one here. All right. So let's get back to the terminal and then we are going to call Bandage. So this is how we call Bandage. All right, so let's clear the screen and then let's do an ls into the assembly directory again and then proceed. So we are now going to visualize the assembly by using this assembly graph here. So we will use Bandage. So to visualize using Bandage, we have to first call Bandage. So if you installed Bandage using Mamba or Conda or micromamba, then you say Bandage, you call it like this, and then you execute the command. So let's execute this command. All right, Bandage has been opened. So we have to load our assembly graph. So to do that, we come to this section here, come to File, and then click on Load Graph. And then you will go and locate your assembly graph. So in the assembly directory, you need to locate assembly_graph.gfa, this one here. So open it. And after opening, you come to this side, the left side here, and then click on Draw Graph. So click it, and then it will be displayed for you. All right, the assembly graph has been drawn for us. Now, what you should notice is that the drawing that you see here can differ from one machine to the other, and even in the same tool, whenever you click on Draw, the diagram will differ a bit. It will change a bit, but it's basically the same. I think there's a bit of randomness in how Bandage draws the graph, so that is something to be expected, so don't worry. All right, now let's proceed. So let's take a look at the glossary page and look at what was done after the assembly graph was drawn. Let's go back. Yeah, so we have that here. So we have this, and we also have this, and that's what we are also seeing here in our graph. So we have this, and then we also have this, and we also have these ones which I wouldn't focus on. I'll rather focus on this one for now. All right, so let's proceed. Let's do something. So remember when we displayed the assembly info.txt, we had some information coming up. Let's just do that again. I'll do that here. So I'll just display it again. I'll say cat assembly/assembly_info.txt. So we had them here. So we have the length, we also have coverage, we also have the context here, by the way. So we are going to focus on just this one here. Look at the length. So if you take a look at the length we have here and compared to the genome length, we see some similarity. All right, now let's go back. Let's go back to the glossary page. So we have some questions here. It says, how many contexts do you have? We had, or let me say, I had 3. What is the coverage of the longest context? And we also have the length of your longest context. And does this feel like potentially an MRSA genome? Let's look at the solution. So with the answers here, it says 2. That's the number of contexts. That is what was displayed here. But on my PC, I had 3. What is the coverage of the longest context? 181. So that is here. That's the longest context by length. It's 181. But for us, or let me say for me, the coverage was what? 198. That's what I have here. All right. Now let's go back to that question. It says, what is the length of the longest context or your longest context? So here it's 2.9 MB. And that is what we also have here. 2.9 MB, that's the longest context. Then let's take a look at the last question. It says, does this feel like potential an MRSA genome? Yeah. And the answer is yes, because we have a 2.9 MB genome, which is approximately the size of an MRSA genome. That's the Staphylococcus aureus. And that's what I just made mention of. So, and then there's also a 1 small potential plasmid genome. So that one is this one. That's what we also had here. This one here. That's what it is. All right. So let's just go back to the glossary page. So basically, these things will also help you to understand your data and combine with the biology of the organism. The information you already have, it's also give you the confidence that indeed the data you have is okay and you are using the right methods and then the right tools for the work. So it's important to always do some of these things as you go along and it also enables you to reproduce the tutorial, or let me say the work. So doing these things helps you to get a workflow or a pipeline that can be reproduced, because at the end of the day, if you have your results and it's not reproducible, then it becomes a challenge. And so it's always important to do these things, and it also gives you the confidence. So with what we have here, if let's say the quantic size, the longest one, or let's say any of them, if we did not have a size which was around this, and let's say we had, let's say 1.2 or let's say 3 point something, then we can start asking other questions, because if indeed we are dealing with an MRSA genome, then we should have this number. So anything other than this number, or anything not close to this number, should raise suspicions. Okay, and the only way you can find out, I repeat, the only way you can find out is to do some of these quality control steps to be able to get them and then proceed. So with what we have done so far, we are confident because we have one quantic or one sequence with this genome size, and that's indicates that yes, indeed, we can proceed with the work. So we can even separate or extract that quantic from the others and then use that for a downstream analysis. So that's how it's done. But of course, for this tutorial, we are not going to do any extraction, by the way. Let's check this. So with the assembly graph that you have, if you display it, it's possible to annotate. So you can add your lengths. I think I should have showed that. So you can add the length here. So now the length, when you check it, you will see this one has a 2.9 MB. There's the quantic. You also have this one. You also have this, and we also have this as well. So that's it. You can add a name. You can also add your coverage. So remember, we had 198 on the table. Let's check for that quantic. It was 198. So that is what has been displayed here. That's for the depth. So if you don't see coverage and you see depth here, it's the same thing. So take note of that. Yeah, you can also do other stuff, but that's beyond the scope of this tutorial. So you can also export to an image file. So you can come to File. I repeat, let me just go over. You can come to File, and then you can save the image, current view, entire scene. Let's just say current view. So you can click it, and then you'll be asked to save. So you can save it anywhere you want, whether MRSA or any directory. I'll just save it here in the same directory as the assembly graph, and I can check. Let's go and check. So this is it. You can open it, you can put this in your reports, or you can also decide to uncheck the length, coverage, and name, and then maintain just the graphs themselves, and then use it for whatever you want to do. So that's also up to you. All right. We'll proceed to do a QC. So what we are going to do next is to run QUAST. So QUAST is a tool that is also used to evaluate genome assembly. So to provide you with some metrics, and this metrics can also help you to evaluate your assembly. So at the end of the day, we are going to get something like this. So let's get back to the terminal. So we are going to run QUAST and then get some quality metrics to evaluate our genome assembly. All right. So we are back to the terminal. So let's clear the screen and continue. All right, so let's do an ls to take a look at our files again. We will ls into the assembly directory. So I'll say ls assembly to take a look at my file. So this is going to be my input file. So I'm going to run QUAST and supply it with this file because I'm going to evaluate this assembly. All right, so to run QUAST, if you install using Mamba, then it has to be this way, QUAST, and then you supply it with the options, including your input file. There are other ways that can be used to install QUAST, and those ones require that when running QUAST, you say QUAST.py. So you need to know how you install it, and you can use the appropriate command. All right, so I installed using Mamba, so I'll use the first approach I indicated. All right, so to evaluate a genome assembly such as this with QUAST, you say QUAST, and then I will indicate my output directory. So I'll say -o, and I'll say QC_assembly. So this will be my output directory. So the output directory here will contain the output files. I'm also going to indicate the number of threads. I'll say --threads. I'll say 4. Now the threads, you can use more than 4 if your PC supports it. So take note. Now I am going to indicate the input file, which is the assembly. So that's this one here. So I'll say assembly/assembly .first A. So this is my assembly file. All right. Now with QUAST, it's possible to also specify a reference genome. So when you do that, you also get some mapping statistics. And it's also possible to specify more than one genome assembly. So you can do that also. So there's a command. So with this command, if you have your own data, you can simply replace this one here with your data, and then you'll be fine. All right. So let's proceed. So if you also have a challenge with running QUAST, you can always let me know, and then we can have a session together. All right. So let's execute this command. So once we do that, QUAST will perform the analysis and give us the outputs. All right. So everything has been done for us. Now let's take a look at the files. So let's do an ls, and we are going to find a directory called QC_assembly. So let's ls into it. We are going to find files here, a lot of files. Let's say a couple of files. So with QUAST, it gives us reports in different file formats. So we have an HTML, which is here. We also have a PDF. We also have text files with information. So it depends on what you want, but everything has been summarized and placed in the HTML and then the PDF. And we also have this one here. So for this tutorial, we are going to look at this one. If it's necessary, we will take a look at the PDF as well. All right, so let's open the files with our file manager. All right, so these are the files. We have all of them here. All right, so let's start with the PDF. So I'll open it now. Yeah, so we have the analysis results here. So you can scroll through and then check whatever you want. But I will use the HTML. So I will open that using my browser. So there's a report.html which you can open. So let's open that using our browser. All right, the report has been opened. So let's enlarge the display. All right. So we have the measurements here, and we also have some plots here. So you can combine both to make meaning or to make sense out of your data. All right. So with QUAST, it performs statistics without reference. So those measurements, they are done regardless of whether you supply it with a reference sequence or not. And we also have some measurement that can be done, or that are done if you supply QUAST with a reference sequence. For example, mapping. So because we did not supply any reference sequence, we don't have the statistics with reference being displayed for us because it was not done. We also have our file here, our sample here. So if you are supplied QUAST with more than one file, then each of the files will have a separate column, and then the respective values will be placed under them. All right, so let's take a look at the discussion that was done on the Galileo tutorial page and then compare with what we have. All right, so let's go down. So this is the report that they got. And then we have some discussions and some statements here. So here it says one can see there are two contigs that has been indicated here. So this one, let's do a quick discussion on that. So with QUAST, this first number of contigs here, whatever number you see here, those are contigs that have their size greater or equal to 500 BP. That has been indicated here. And so sometimes maybe you perform some Linux commands to get your total number of contigs, but then you find that the number you had is different from what is here. That means that some of your contigs had their sizes less than 500 BP. So that's what you should note. All right, so if you want the total contigs that are in your file, the original file, then you have to check this one here, contigs greater or equal to 0 BP. Then that one will give you everything in the original file. So I thought I'll bring this to your attention. All right. So for us, we have 3 contigs and all of them have their sizes greater or equal to 500 BP. So that's fine. So let's scroll down. The largest contig is 2927. That's what was reported here in this tutorial. And then comparing to this one, the sizes are almost the same. So yes, 2927008. And then for this paper, the one that reported this isolates, they had 2976370. So they are almost the same. For us, we had 2907. That's what we had here. So it's also slightly different, but I would say it's almost the same. So there could be a number of reasons. One possible reason would be the Flye assembly. Remember, we had this note here telling us that the results can differ from this example here. Okay, because Flye can differ a bit per assembly. So that could also have affected our work. And then the other thing is that for genome assembly using Flye, it's possible to do polishing while the assembly is going on. So Flye allows you to specify an option for polishing. And so there's a default value, but it's possible to increase it. And so maybe if we had increased that number, we could have gotten something similar, or we could have gotten the same as what was reported here. So that is something that you also need to take into consideration. So if you have some of these measurements done and you compare to your reference genome, you can always try to improve it by doing polishing, and then just compare the polished one to the original one and then see which one is of higher quality or which one is closer to the reference sequence. Then you can use that for a downstream analysis. So these are things you always have to do. All right, so the largest contig here has this one. So this could mean that the contig is the total chromosomal DNA of the isolates, S Aureus. And so linking that to Aureus, we also have this. So the conclusion is the same as what is given here, because we also have something similar. And so you can try to improve it, or you can use that for the downstream analysis. So since it's not exactly the same, there are a number of factors which I've made mention of, and there could be others. And so the most important thing is to make sure you have something very close to what you have, and then maybe you can investigate why there's that difference later on. So that's it. Another thing you can also use to compare is the GC content. So let's take a look at this question. It says, what is the GC content? And it says the GC content for our assembly is 32.73%. That is what was reported on this page. For mine, GC content is 32.92. Okay. So with the MRSA isolates, GC content is 32.89%. Ours is 32.92%, which is slightly above. So they are almost the same. They are almost the same. That's the difference is not that much. And so this also means that the length and the GC contents of our assembly is good because the values are close to the MRSA, which is being used as the reference sequence. So that's it. So that is it for the QUAST quality control tool. And so what we are going to do next is to move on to our next activity. So the next activity, we are going to identify antimicrobial resistance genes. So for that, we are going to use the tool STARAMR. So we will go to the terminal and then run STARAMR. I've already covered an entire tutorial on that, so you can also check the description box for that tutorial. All right, so let's proceed. Let's go back to the terminal. So let's clear the screen and continue. All right, so I'll do an ls and I'll ls into the assembly directory to take a look at the file. All right, so this is going to be my input file. So this is our assembly data, which has the context. So we are going to identify antimicrobial resistance genes in this data. So I'll use STARAMR. So to identify antimicrobial resistance genes using STARAMR, we say STARAMR and we say search. And then I'll indicate my output directory. So that will be -o. Then I'll specify the name. So it will be the sample name, and I'll add _STARAMR. So this directory will contain the output files. Now let's specify the input file, which is assembly.fasta. So that will be assembly/assembly.fasta. If you are going to use your own input file, then you can just change the output directory name and then the file name here. All right, so that's what we are going to do. Now, STARAMR, in addition to identify antimicrobial resistant genes, also identifies plasmids, and it also tries to do some multilocal sequence typing as well. It does that, so you get the MLST results as well. There are other files it generates, and I've covered an entire tutorial on that, so check the description box, you'll find a link to that tutorial. Okay, so let's continue. So let's execute this command to run STARAMR. All right, so STARAMR is being executed. All right, so in some cases, you may identify this error message here, but don't worry, we are going to resolve it. So this is happening because the current STARAMR version I'm using, one of the commands is not compatible with the pandas version that was also installed. So pandas is a Python library. So what we are going to do is to resolve this issue. So we resolve it by first removing that pandas version and then installing an earlier one, an older version. So that's what we are going to do next. So let's clear the screen and continue. All right, so I'll use pip to remove the pandas library, the current one. So I'll say pip uninstall pandas. I'll confirm the uninstallation. All right, so pandas has been removed. We are now going to reinstall, but this time we will use an older version. So I'll say pip install pandas equals 1.5.3. This worked for me, so I'll use this version. So I'll execute this command, and then pip will install that version for me. All right, that version has been installed, so we can proceed. Now, before I rerun STARAMR, I will have to remove this directory because I'll use the same name, so I'll remove it here. So I'll say rm-fr, and then I'll remove it. you should be careful when using the rm command, because once you execute it, there's no coming back. Your files are gone, so be careful. All right, so let's proceed. I'll just redo that again. All right, so let's rerun STARAMR. So I'll say STARAMR search -o, and then I indicate my output directory, and then I'll specify my assembly file. All right, so let's execute this command. So this time, STARAMR will be able to do its work. All right, so everything has been done. So with this one, you can ignore it. That's fine. It's nothing to get worried about. Okay, so we have been able to successfully execute STARAMR. I repeat, we have been able to successfully execute STARAMR. So we have the outputs. Let's take a look at them. So let's do an ls. We will have our output directory being displayed for us. So this one here. So let's ls into it. And we are going to find our files there. So we have a couple of files. Now, these files, a description of each of these files can be found on the STARAMR page. I will leave that link in the description box. Now, I've also covered an entire tutorial on that as well, so you can check the description box for all those materials. All right, so we are going to open some of these files, by the way, so I'll do that using my file manager. So let's go and then take a look at the files. All right, so these are the output files. So for this tutorial, I'll focus on just this one and this one and this and this. So you can open any of them with a spreadsheet software. So let's take a look at the REST finder. So this one has information about the antimicrobial agents. So we can open it with a spreadsheet software and then look at what we have. So each row is a hit, a record. Each row represents an identified AMR genes. So we have the isolate ID here. It will be the same name because we specified just one file. And so all the hits are in that file. And we have the gene name. We also have predicted phenotype, percentage identity, overlap, etc. We also have contig. So here, because we have more than one contig, if a gene is identified on a particular contig, then the name of the contig will be indicated here. So this will let you know which contig that gene was found on, that AMR gene was found on. So here we have contig 2. We also have contig 1. So here it tells us that contig 1 and 2 were the ones which had the AMR genes, the antimicrobial genes. So we have the details here. All right, we also have accession number. We have start, end, accession number, sequence, etc. So you can look the gene up using the accession number. Let's take a look at a discussion on the Galaxy tutorial page here. So the Galaxy tutorial page also gives the files and it gives a description. So you can read that or visit the developers GitHub page. I mean the STARAMR developers GitHub page. All right. There's also a discussion here about multi-locale sequence typing. So with the multi-locale sequence typing, the Galaxy tutorial page makes us know that using error-rich sequence data like Nanopore, that's long reads, then it's going to be challenging if you try to perform MLST using such kind of data. So you are better off doing it using a short read data, i.e. Illumina. Okay, so that's what this statement means, but I would like you to read it yourself and then try to get the information. And we also have plasmid and then antimicrobial genes. So we have the information here. We also have this one here. It says multiple rep sequences are located on the second contig. So the second contig, ours may be different because we have a slightly different results. So what I have to do is check if you can find the rep sequences. So here, the rep sequences here are found on plasmids. And so what this indicates is that since there are rep sequences, that means that our sequence data likely has plasmid or plasmids. Let's take a look at our file. We will look at the plasmid finder file. So this has information about plasmid. So let's open this also. So if you open it, you will get information. So we have rep sequences. That's what the tutorial is referring to. So we have them here. We have the contigs. So yeah, we can see that rep sequences have been found in all the contigs. So contig 2, 4, and then 1. They all have rep sequences. And then we have the identity here. So identity here is less than 100. So for me, if it's less than 100, I'll want to do some other checks to make sure that these are indeed plasmids. But I mean, once you have these ones here, and then the thresholds to, you decide the threshold. So for me, I'll go for 100% identity, but it depends on what you want. You can also look at having a threshold less than 100 and then decide if the hit you have is indeed a plasmid or not. Okay, so that's what we have here. And then we also have the session IDs to let you know them. So you can just set the session IDs to check the information about that. All right, so that's what we have. So we also have multiple resistant genes can be found on both contig 1 and 2. For us, we found in all the 3 contigs. And then the session number, I've talked about it. So you can use that session number to read more about the gene or the sequence or that hits. So you can do that. And we also have this database here. So it says we can get more information about our antimicrobial genes by visiting this page here, this database. So if you want more information, just visit that page. The link is here. Just open it. And then when you open it, you can do your search or do some analysis here as well. All right, so that is it. And we also have some questions here which I would like you to answer. One is about the MEC-A. And then we also have other questions here which I would like you to check and then see. All right, so that is it. So I'll move on to the next activity, and that is to perform gene annotation using PROKKA. That's what we are going to do next. So again, we will use the Flye assembly output for this exercise. So let's do that now. Let's go back to the terminal and continue. All right, let's ls into the assembly directory. So what we are going to do next is to annotate the sequences that can be found in this file. So this file here has contigs. So we are going to annotate the sequences using PROKKA. So we will say PROKKA, and then we will specify some options. So the options, we will include those stated in the Galaxy tutorial. So let's just check them. So there's the section for annotation. So we are using PROKKA. So we have to specify the input file, which is the Flye assembly output. We have that. We also need to indicate genus, Staphylococcus, species, Aureus, and then kingdom bacteria. So we will add these. And then we will include other options as well. So let's get back to the terminal. All right, so I have typed PROKKA and I'll say -- genus, that'll be Staphylococcus, and then species, that'll be Aureus, and then kingdom, that'll be bacteria, and then I'll indicate CPUs. I'll say 4. With the CPUs, you can increase it if you have more capacity. Now let's add prefix. So I'll say dash prefix space, and then I'll indicate the sample name, and I'll say underscore PROKKA. So what will happen is that PROKKA will create a directory that has this name, and in this directory you will have your output file. So the output files will also have the same name here, in addition to their respective file extensions. All right, now let's indicate the input file. So that will be assembly / assembly.first-a. So now let's execute this command. All right, so PROKKA has started this way. So let's wait for everything to get done for us. All right, PROKKA has completed its work, so we can proceed. So let's take a look at the output files. So let's do an ls, and we are going to find this directory. This has the output files. So we'll ls into it. We are going to find our files here. So that's how we do it. Now let's proceed. So I will focus on a few of the files. I will start with this one here. So this file, you can use it to take a peek into your data to look at the features. Example, predicted genes or CDS and all that stuff. So let's display the contents here. We will use the head command. So I'll say head, and then I'll indicate the file, which is a TSV file, by the way. So TSV. So because it's a TSV file, the entries here, the data is tab separated. That means it contains tab separated values. So you can open this file here using a spreadsheet software. So take notes. But now I'm using bash commands to get the contents. All right, so I'll do a head to display some of the contents. So we have locus tab, F type, length, gene, etc. So these are the features. So features, CDS, gene, RNA, etc. So those ones are here. So they are tab separated values. So this is column 1, this is column 2, column 3, up to the end here. All right. So for me, what I'm interested in is to get a summary. How many CDS do we have? How many tRNA do we have? How many genes do we have? So I'll do this. I'll just get the features here. Let's just do this again. I'll do the ls again. So I'll get the features. That's what I want. So I'll use the cat command to do that. So I'll say cat-f2 because I want the second column, which is the F type. And then I'll specify the file, which is the TSV file. And then I'll do a head to get a few lines. So I have them here. So this is what I have. But with what I have, this F type here is not part, there's a header. So I can still remove the header before getting these ones. So I can do it this way. So with the same command here, I can say tail -n +2. So +2 means that you have to start from the second line. That's what it means. And I can now do a head. So this will remove that header for me. Okay, now let's expand it. So we have them, but I would like to count how many each has. How many each of them occurs, or how many features are assigned to or belong to CDS or RNA or etc. We are going to do that. So let's do this. So let's do this. I'll reissue the same command here, but this time when I come here, I'll say sort and I'll say unique. So let's execute this command. So this will give me just the features. That's what I want for now. So CDS, RNA, tmRNA, tRNA. So here we see that there are no genes. No genes have been predicted. And so that's what we have. Of course, the data we are dealing with is probably a downsampled one, and so I wouldn't be surprised if we have these things happening. Of course, we can go further into it. All right. So this is what we have for the features. Okay. So yeah, for the data we have here, there are no genes. I would have expected to see genes also here. We have CDS, we should have genes, but that's what we have anyway. Maybe the data is downsampled, or I'm not really sure, but this is what I have, by the way, so I'll just go with it. So this is a practice data, but if you are dealing with whole genome sequence for bacteria, organisms, or bacterial isolates, then you should expect that PROKKA also gives you features that are genes, so take note of that. All right, so we can count them. So using the same command here, I'll just add -c, and that'll give me the counts. So I'll execute this command, I'll get a count. So here it tells me 5114 CDS. That means that 5,114 of the features are CDS. We also have 16 RNAs, there's 61 tRNA. So that's what we have here. So this is a simple command to get you started with exploring the data. All right. So you can still view with your file manager, by the way. So try your hands and let's see what you have. Now let's go to the gallery tutorial page to see what was done next. All right. So here the gallery tutorial page only indicated the GFF3 format. So that is the only output file that it's needed for the rest of the analysis, but we generated all of them, so it's fine. We will proceed. So what was done next is to select some records from the STARAMR results that is here. And so we're also going to do the same. So it tells us that we need to select lines with a decimal value followed by a tab character. Okay, because we want to filter out some entries without identity value. We need to filter them out from the STARAMR result, the detailed summary file. Let's take a look at that file first. So if you come to the STARAMR folder, if you get there, you look at this file here. Let's open it. We have some entries here. So what the tutorial is trying to do, or hopes to achieve, that instruction is that we select only records with identity values. That's the presenter identity. So here you can see there's one here which doesn't have. So we need to get a command that will extract all these ones for us and then leave this one. So we are going to do everything on the command line. With a spreadsheet software, it's easy to do it, but we will still do everything on the command line, because ideally, if you're working on a Linux system, then you should be able to learn how to do some of these things there. They become very, very crucial. And they are very handy, by the way. So let's get back to the command line. All right, so I'll proceed. So I'll clear the screen. I'll ls into the STARAMR directory to find the file that we need. So we need this one here. That is what we need. So we are going to do the extraction here. So we are going to use a grep command to do this. So let's continue. All right. So I am going to use the pattern that was indicated in the gallery tutorial page. So let's go to the page. All right, so we are here. So we are going to select lines that match an expression, and the expression is this one. So I'll copy it. And then with the expression, we are supposed to select lines from this file, the detailed _ summary.csv. So this is a STARAMR output file. So let's get back to the terminal and then extract those lines. All right, so I'll use grep and I'll say -p, and then the -p here means I'm going to use the Perl style. I'll bring my quotes and then I'll paste the pattern there, and then I'll bring another quote again. They are all single quotes, and then I'll specify the file. So this will be the final command. We need to redirect the output to a file, but for now, let's just display it. So let's hit the enter button, and then we have the results here. So we want to redirect everything to an output file. So I'll reissue the command, and then I'll bring my greater than symbol, and then I'll say STARAMR dot filtered dot CSV, like this, STARAMR dot filtered dot CSV, and then I'll execute this command. So if I do an ls, I'm going to find it here. I can still do a cut here to display the contents, and I have them here. So this is what I have. All right. So we can also do a WC-L to count the lines. We have 17 of them. So we have 17 entries. That's what we have. All right. So let's proceed. All right. So let's take a look at what we are supposed to do next. Let's go back to the Galaxy tutorial page. So after generating the output file, after using the grep command to obtain the needed information which have been saved here, we need to convert the outputs, or let's just say we need to generate a GFF file from that output. All right, and so that means we need to use this particular file here. This will be the input file. So we will extract the information from this outputs and then place them in a GFF file. So we are going to get a GFF file. So in other words, we are going to convert this output here to a GFF format. So that's what we are going to do next. So the instructions are here, but I have written a Python script that we can use to generate that GFF file. And so you don't need to do any heavy lifting. Just go to this GitHub page here. This is a Python script. I'll leave the link to this page in the description box. So when you are here to download, you come to raw, this one here, click on raw, and then when you are here, right click and then go to save pages, and then you can save it wherever you want to. Okay, another way to also save it is to visit the same page here, and then when you come to raw, right click it and then go to copy link, and then you can move to the terminal and then use wget. So I will do that. So I'll say wget and then I'll paste the link there and I'll execute this command. So this will download the file for me. So now it's done. If I do an ls, I'm going to find a file there. So this is it. This is a Python script. So I'm going to run the script. So I've already entered all the necessary information in there, and so all that I have to do is to execute it. So you need to also make sure that this file here, we have generated it, but make sure it's in your current working directory and it has the same name. If you're using a different file, then you have to change it in the script. So that one, it's up to you, but for now, just make sure that all the information that has been specified in this script here, you have them. That's all that you need. All right, so I am going to execute the script. So I'll say Python and then I'll call the script, that's STARAMR _2 _gff.py. So let's execute this command. So after executing it, if you do an ls, you are going to find this file here. This is the STARAMR hits.gff. So this is the GFF file. You can do an exploration. You can say head STARAMR hits.gff just to look at the contents. So that's what you have. So I tried to make it generate the same or almost the same output as what can be generated from the Galaxy platform. So the script here is not so elegant, but it's going to work for us. So just make sure that you run it as it is. You can also experiment with other tools to see, and then just use whatever you are comfortable with. But this script should work for us for the most part. All right. So that has been done. Let's go back to the tutorial page. So we now have our GFF file. So after generating the GFF file, we need to also do some mapping here. So that's what we are going to do next. So with the mapping, we are going to map the reads that can be found in this file, the filtered reads. We are going to map them to the output of the Flye assembler. So here we are referring to the assembly context. So the assembly context become our reference sequence. We are going to use minimap2 to do this. So if you want to read more about minimap2, you can visit the developers GitHub page, which is here. I'll leave the link to this page in the description box. So we are going to use this command here because we are dealing with Oxford Nanopore reads. So we will use this command. We will do some slight modification here, but for the most part, this is the syntax. All right. So let's get back to the terminal and then continue. So let's clear the screen and then continue. So let's do an ls. So this is our FASTQ file. So we are going to map the reads in this file to our assembly context, which can be found here. So let's ls into the assembly directory to take a look at our file. So this is the file. This becomes our reference sequence. What I'm going to do next is to make a copy of this file. So I will say make the ref, and then I'll copy the file into it. So I'll say cp assembly / assembly.fasta, and then I'll say ref. I'm making a copy because I want to avoid accidentally modifying the original file. So that's it. All right. Now before we run minimap2, let's also do this. Let's also create a directory that we will store the BAM file. So I'll say make the mapping, and then we are ready. So let's also ls into the ref directory. All right. So now we have our FASTQ file here, which has the reads. We also have our reference sequence here. So we are going to use minimap2 to map the reads to this sequence. All right, so let's run minimap2. So I'll say minimap2, now say -ax, now say map-ont, and I'll say -t, that's the number of threads. So here I'll use 6, but you can use 8, 10, or whatever you have available. If you have more than 10, you can specify it. If you are working on a Linux cluster, an HP system, you can have more CPU cores, so you can increase this number to 20 or whatever, but I'll use 6 because I am working on my normal laptop. All right. So I'll also indicate the ref. So I'll say ref / assembly.fasta. That becomes my reference sequence, and then I'll indicate the reads. So the reads here, we'll use this file here. So that'll be this .filter.fastq. All right. Now this is the command for mapping, but we also need to do this. We also need to sort the BAM files. The BAM file that we will generate, the output, we have to sort it. So I'll bring another command here, and then I'll say some tools sorts. And I'll say -o, and I'll say mapping /, and then I will give the base name here, this sample name here. I'll paste it, and I'll say dot .bam. All right, you can also use .sorted.bam. That's also fine. So it depends on what you want at the end, but I'm using .bam because I'm going to generate just a single BAM file. But anyway, let's use .sorted.bam. I think that's better so that we have a description of the file. So we will say this .sorted.bam. So this becomes our final command. So we have our minimap2 command, and then we use some tools to sort the resulting outputs. So let's execute this command. All right, so now mapping has been done. So let's wait for everything to get done for us. All right, minimap2 has completed its work. So let's take a look at the outputs. So let's ls into the mapping directory. Let's do an ls first. So we have our mapping directory, which has the output. That's the BAM file. So I'll say ls mapping, and I'm going to see my file here. Now, what I'm going to do next is to index this BAM file. So I'll say some tools index, and I'll say mapping, and then I'll indicate the BAM file. So this will index the BAM file for me. All right. Now we have successfully indexed the BAM file, so we are ready for the next activity. So let's clear the screen and continue. All right, now let's do an ls again. So we have our outputs. Now what we are going to do next is to visualize our output. All right, so we will use 4 input files here. Let's go back to the gallery tutorial page and then look at what was done. So with the visualization, JBROWSE was used. All right, we will configure JBROWSE very soon. So JBROWSE was used, and then the input files that were used were number 1, the Flye assembly output, and then number 2, the PROKKA output, one of the files, that's the GFF file. All right, that's also here. And then number 3, we also have the STARAMR file. We converted the filtered file to a GFF. So we are also going to use the GFF file that we generated. And then we also specify the BAM file that we generated when we perform the mapping with minmap2. So these are the 4 input files we use. All right, so that's basically it. Let's go back to our terminal and then let's take a look at these files. So we have our assembly. Let's ls into assembly. This is the first input, and then we also have PROKKA. So let's ls into PROKKA as a PROKKA output, by the way. And then we are going to use the GFF, which is here, this one here. That's what we are going to use. And then the third file is this one, the STARAMR underscore hits. Let's just do an ls here to simplify stuff. So the STARAMR hits.gff, the one we generated when we ran the Python script. All right. So that is also going to be used. And then we are going to also use the BAM file that we generated. So let's ls into mapping to see that file. So we will use this file. So these are the 4 files that we will use. All right. Now, before we call JBROWSE, we need to set it up. And so what we have to do is to download the JBROWSE executable. All right. So let's go to the JBROWSE page and then download it. So there's a page, and I'll leave the link to this page in the description box. So when you are here, just come to this site. There's a download button. If you don't see it here, just click on this download icon here, and then you be sent to the download page. So here you will download the one that you want. So for this tutorial, we are going to use the command line version, so we will use this one. All right. So I'll right click, and then I'll go to copy link. and then I'll move to the terminal to download it. You can also click to download, but I will do it on the terminal. So that's why I copied the link. So I'll just do it again. I'll right click and I'll copy the link. This is the download link. And then I'll move to the terminal. So on the terminal, I'll just clear the screen and do this. I'll say wget, and then I'll paste the executable there. All right, so let's execute this command. All right, so once we do that, JBROWSE will be downloaded for us. So let's wait for the executable to be downloaded for us. Okay, the JBROWSE executable has been downloaded. So let's check it out. Let's do an ls and we are going to find it here. All right, so there's it. Now for this tutorial, we are going to use it as it is. We will do a little modification, but then I have a separate tutorial that shows you how to download it and then configure it so that you can use it anywhere on your system. All right, so I would advise you to check that video out if you are going to be using JBROWSE on a regular basis. All right, so let's continue. So with what we have now, we will rename the JBROWSE file to make it shorter. So I'll say mv, and then I'll specify the file. And I'll say JBROWSE like this. So this will rename the file for me. So I'll say ls and I'm going to find it here. That's what I have. What I'm going to do next is to add execution rights. So I'll say chmod +x and I'll say JBROWSE. All right, so now that has been done. So if I do an ls, I'll see the color change. So depend on your system, the system that you are using, the color may change or not. But once you execute this command, everything is fine. So don't worry if you don't see any color change in the file, the name. All right, so that's fine. So that's how we set up JBROWSE. So it's time to do the visualization. All right, so we are now going to call JBROWSE. We are going to open it. All right, I'll do it here. So to open JBROWSE, I'll say // and I'll say JBROWSE. So I'll execute this command. So once I do that, JBROWSE is going to be opened for me. All right, so now we can start. All right, so once you have JBROWSE open, this is the interface. So we will come to this side, the left side here, which says launch new session. So come to this side, open sequence file. So click it, and then you have this coming up. All right. So when we are here, we will first give a name. Okay, so here it says assembly name, so we have to give it a name. So I'll use the sample name for our data. All right, now we need to load the reference sequence. So in this case, the reference sequence becomes our assembly data. All right, so here we will use this other one. Let's click this arrow here, and then select first A adapter. Now go and locate the assembly file. So click choose file, and then locate the file. So for this tutorial, we made a copy of it. Okay, so the copy can be found in the ref directory, but you don't need to do that. You can also use this one. So let's just use assembly. All right, so click assembly and then select the assembly.first-A. But ideally, what you should do is to make a copy of this file and then use it so that you don't accidentally modify the original file. That's what I recommend. All right, so just make sure to do that. Okay, so I will use the ref one because it's the same. All right, so I'll just click ref and then click on assembly.first-A. So that is what I'll open now. So this is our reference sequence. All right, and then I'll click on submit, this one here. And then after that, I'll come to this side, launch view. All right, here you'll see linear genome view. That is what we'll use. If it's not linear genome view, just click and select linear genome view. Later, you can experiment with the other ones, but let's use linear genome view. So click on launch view after you have selected linear genome view. All right, so when you click, you have this coming up to let you select a config, but we are going to use everything. All right, so here you can select config 1, 2, 4. These are the sequences in the reference sequence that we selected, the assembly, okay, the assembly file, but we want to show all regions. So we will leave everything here and then come to this side, show all regions in assembly. Later on, we can select the one that we want. So click show all regions in assembly, and then you will have this coming up. So what we are going to do next is to add the other files. Now we have loaded the assembly.fasta. We need to add the PROKKA, the STARAMR filter GFF, and we also need to load the BAM file. So come to this side, open track selector, and then click it. So when you click it, you will have this coming up. All right. So here, come to this side, the lower side here, and then click on the plus, and then click on add track. And then you can select the file. So we will start with the PROKKA GFF file. So click on this one. When you come to this side, click on file, this one here, make sure it's highlighted, and then come to choose file. Click choose file, and then select the PROKKA GFF. That's a here, mine is here. So open it, and then go to next. And then here, you can choose to rename it. There's a track name. I will just remove the .gff and I'll use this name here, right? You can also use PROKKA or whatever, that's fine, but I will leave it as it is. And then come to add, click it, and then you have it there. There are still things we will do, so don't worry. Now, when you come to this side, running jobs, click on this plus, and then that one will be removed or hidden, and then you'll come again. For now, don't worry about these things, we will come back to them. Let's add the other file. So click the plus, click on add track, click on file, choose file, and this time we are going to select the STARAMR filtered GFF. Okay, this one here, STARAMR_hits.gff, this one here. So click it, and then come to next. Here, let's also remove the .gff. Let's leave it as it is. And then scroll down, come to add. All right, that is also done. So now we see some stuffs coming up. Now let's close and add a BAM file. So come again, plus, add track, file, choose file, and this time let's select the BAM file. So that is in the mapping directory. So this one here. So open it, or select, and then go to next. Here, let's just leave it as it is. Yeah, you can always change it as well, but that's fine. So I'll now do add, and that is also done. All right, so you may have this coming up which says too much data, but these are things you can resolve later on. It's a matter of you doing some zooming and all that stuff. So you can do that later on. So the focus is to look at these stuffs here. So let's take a look at something. So here, the tracks here, you can check the one that you want displayed for you. All right, so here, if I check everything, that means that I want everything, all the information in the various tracks, the input files, all of them should be displayed for me here. All right, that's what you have. But for this tutorial, we will focus on just two of them. I'll focus on the PROKKA and then the STARAMR hits. So I will remove this and this for now. I will come back to them later. Okay, so this is the STARAMR hit. So you can see we have the session IDs, okay, here for the hits. Now let's even look at the spreadsheet first. So this is the filtered one. So let's open it. and then we have our files here. So we can see some of the accession IDs. Let me enlarge it a bit. You can see them. This is okay. Let's look at the JBROWSE. Do I have them here? Let's see. Let's check. Okay, so we have our JBROWSE. So we can take a look at the STARAMR heads. You can see these ones here. You can also scroll down here and then see them here. All right, so we see some of them here. You can click and then check where they are. So the information that is in the GFF will be shown for us. All right, so we have, let's take a look at M19465. We can check it here. M19, that is it. All right, so we have that here. Okay, now let's proceed. Let's go back. Now what we are also going to do is to also enable the PROKKA annotations. I think it's already there, but we have to just reload it. So I'll just click on the force load to load it again. Yeah, perfect. So we have them here. Okay, let's do this. Let's select a different config. Yeah, so you can select the different configs and then just look at what you have. Okay, so here it seems there's too much information in config 1, but that's fine. We will just look at the others. It's fine. Okay, so this is config 2. That's all. This is config 4. And then for config 1, we have too much information. So if it's too much information, you can just zoom out and then just pause to reload and then just see. All right, those things do happen. So we will just have to find a way to manage them. Okay, so let's go to the Galaxy tutorial page. Let's take a look at the discussion. Yeah, so we will have something like this. Of course, the picture here, ours is a bit different because we had the config 1 here. I mean, not necessarily the same as the config 1 in our file. All right, so that means the config 1 that we have, you may not see these annotations in there. All right, so let's do this. So let's take a look at something else here. You can search for features. All right, so the tutorial page here makes us aware that we can search for features, but of course, searching it in the JBROWSE, the method may be different a bit, but we will use a different approach to do the searching. But let's just go to this section here. I want us to focus on this one here. All right, so it says now we are going to search for AA6, APH2 gene. And we're searching for this blah blah. So this is one of the AMR genes that we got. So the tutorial page here says we can search for it. Now before we search, all right, let's go to the spreadsheet again to search for that AMR gene. So that is here. Let's check. It's this one here. Okay, so here we have the start and end. All right, in this spreadsheet, it's not there, but that's how it's supposed to be. You should have the start and end here. When we use the grep command to filter and get the lines of interest, we did not add a header, but the headers here are the same as the ones in the original file, so you can just do the maths. So here we have the config, we have the start and then the end. All right, so if you take a look at this one here, that AMR gene that the tutorial page makes mention of, this is the start and end. All right, so we have this and this. So here we have 27414, here we have 2592. So this indicates that it's likely to be in the reverse strand. All right, so if you are going to search, you will still need to use the lowest number or the smallest number first, and then the highest number second. And then we also need to indicate the context. So what we are going to do is to search for this particular gene here. All right, so note that we have the PROKKA annotation and we also have the STARAMR hits information. So we are going to search and then see where, the PROKKA annotations where these this gene can be found. So we will need the information from this side here. So I will just make a copy of one of the values. The rest I will just type. I'll just copy this. All right. So let's go and then search for that gene. So that gene can be found on contact 2. All right. So we will have to also change it here. So here we will do this. So we will clear and then start with this. We are starting with the lowest, and then let me just do it again. We will start with this one here. So it's supposed to be contact 2. All right, contact 2, and then let's clear everything and then specify the region. So we paste the smaller value and we say dot dot, and then we bring the other value, which is 27,414. All right, so this is it. So this one is also comma. Let's just add it like this, and then let's hit the enter button. Okay, so when you hit the enter button, you will have the region being displayed for you. So this is it. Okay, so for the PROKKA annotation, we see that region, and then when you slide, you can see the session ID. Okay, here we can see the session ID for the STARAMR head. So it's from this side up to this side. Okay, so in the same region in PROKKA, we see multiple copies of that gene. So we can see them here. We can see this. We can see this. We can see this. All right, so we have all those ones. So we have 3 copies for that gene. All right, so in the Galaxy tutorial page, for the Galaxy tutorial page, they had, let's say the number here is, I think, it's 5. Let's say 1, 2, 3, 4, 5, 6, 7, 8. Let's check the number. I think I saw the number somewhere here. I'll just check. Yeah, so I think it says we have multiple AACA, APHD genes are found within the STARAMR annotated this one here. All right, so this region here. So we have multiple of that gene. That's what we have also seen. So the number that we have is different from what was reported in the Galaxy page. Of course, everything boils down to the assembly. And so the assembly we got determine the output for the PROKKA annotation as well as the other ones. But for the most part, we have been able to reproduce everything here. All right, so with some of this analysis, you may not get the exact same output, but you will get something close. So that will let you know it's reproducible. Some of the tools are a bit stochastic, and so you may not get the same output even if you rerun the tool multiple times. Yeah, so that's about it for now. So that is it for the visualization, and you can also answer this question. It says, did the location of the AAC regions in this correspond to any of the found genes in the PROKKA JBROWSE output? Yes, and that is what we just discussed. That yes, multiple AACA PhD genes are found within the STARAMR annotated this. All right, so the multiple copies are here. So this is the multiple copies. You have this, we have this, we have this. All right, and this is the STARAMR annotated region. Okay, so that same region, we have this and this. You can also check. Let's just go back to that and see. We also have this. You can also do for this other ones to see if there are multiple copies. All right, so you can also do it for the plasmids. also to see where they are, but that's something that I would like you to try and then give me a feedback. All right, so these are cool things that you can do if you know your way around the bioinformatics. So of course, this is a bacterial genome, and so it makes sense that we do all these things to visualize and you'll be able to understand the organism better with respect to the genes of interest or the features of interest or the genomes or the sequence that we are interested in. All right, so these are things you can also do. All right, you can also include the alignment records by clicking it, and then it will be shown here, but there's a lot of information, so it takes time to add that. All right, so we have that here. You can also add a reference sequence, which is here, to give you another visualization, and you can visualize and then make sense out of it. All right, but a detailed discussion of everything here is beyond the scope of this tutorial, so just make sure to do it yourself and then bring me a feedback. All right, so that's what we have for our data. So in this tutorial, we have been able to perform a number of activities to understand the organism better with respect to antimicrobial resistant genes. And so it becomes very easy now to tell your story or write your report based on whatever you have seen here. Of course, this is where the visualizations will come. So doing the visualizations allow you to kind of understand the data better, and then you can proceed with the other analysis. And if you're interested in more bacterial genomics tutorials, then you can check this playlist that's been shown right now.